My name is Johnny Sandoval. I talk about patient customized oligonucleotide therapy for rare genetic disease. Definition: this describes how molecular diagnosis of a rare fatal neurodegenerative condition led to the regional design, testing, and manufacture of medicine as spliced modulating antisense oligonucleotide drug tailored to a particular patient. Rare disease in a great affect approximately 30 million persons in the United States alone.
Number 10. RNA-based therapies.
A code of genetic information. Ribonucleic acid has become a popular target for therapeutics. Interfering with that code gives scientists the ability to intercept a patient's genetic abnormality before it is translated into non-functioning proteins. Antisense nucleotide therapy prevents the production of proteins encoded by a disease and is being explored in several conditions, such as age-related retinitis, familial hypercholesterolemia, and Huntington's disease. RNA interference, a mechanism for gene silencing, is currently being explored for use in cancer and neurological diseases. With RNA interference, RNA molecules, either microRNA, miRNA, or short interfering RNA, siRNA, inhibit gene expression by neutralizing targeted mRNA sequences. FDA approved in 2018, the most successful RNA interference therapy is for the treatment of hereditary transthyretin-mediated amyloidosis. Feeding off the success of DNA-based gene therapy, RNA-based therapy puts a twist on treatment, sparking progress and innovation in therapeutics.
Application. These reports showed a path to personalized treatment for patients with orphan disease. The number of patients affected by each rare disease presents major challenge for drug development. After obtaining rare informed consent from the patient parents, we obtain blood and skin samples for genomic and molecular characterization under an appropriate human subjects research protocol approved by the Institutional Review Board of Boston Hospital.
Therapeutic oligonucleotides are a class of drugs that target messenger RNAs and change protein expression. They can be used to treat a diverse range of diseases. Oligonucleotides are short synthetic polymers of nucleotides that are complementary to specific regions of mRNA that encode disease-related proteins. Therapeutic oligonucleotides are designed to be highly specific for the target RNA to reduce the odds of off-target effects on bystander RNAs. After parenteral administration, the oligonucleotide enters a cell and binds to complementary messenger RNA. Depending on the disease mechanism and the intended outcome, binding of the oligonucleotide to messenger RNA induces a series of events, including a change in the pre-messenger RNA splicing and the destruction of messenger RNA by means of enzymatic cleavage. Alternative splicing of pre-messenger RNA yields different proteins with altered function. Spinal muscular atrophy is caused by mutations in the gene SMN1, resulting in loss of SMN protein function. Small amounts of SMN protein are also produced by the gene SMN2, but most of the SMN2 RNA is degraded, owing to a variant that interacts with the splicing machinery, resulting in exon excision. Nusinersen is an oligonucleotide drug that blocks the excision of the exon. Consequently, SMN2 messenger RNA is processed to include the excluded exon, leading to more stable messenger RNA and the production of functional SMN2-encoded SMN protein. Destruction of the messenger RNA has been used to treat hyperlipidemia. Mipomersen is a single-stranded oligonucleotide that is complementary to RNA encoding apolipoprotein B, a component of LDL cholesterol. Binding of mipomersen to pre-messenger RNA that encodes apolipoprotein B leads to cleavage of the messenger RNA, reducing apolipoprotein B production and therefore hepatic LDL production, even in patients for whom statin therapy has been ineffective. Despite the advantages of oligonucleotide drug therapy, it must clear the same hurdles as other types of drugs, such as safety and achieving effective delivery. Safety issues flagged in clinical trials of oligonucleotide drugs include the activation of pro-inflammatory response and, for certain oligonucleotides, renal accumulation and reduced platelet count. As they are hydrophilic and relatively large, their ability to penetrate cell membranes and hit target sites creates a challenge for effective delivery. Nonetheless, the ability to use genomic information to design drugs to treat diseases once thought to be refractory to drug therapy is encouraging and exciting.
Treatment. There were no serious adverse events. Treatment was conducted under an expanded access investigational clinical protocol, and it has also been accompanied by objective reductions in the frequency and duration of seizures.
If your child had a rare disease with no cure, how far would you go to save them?
Yeah, some families are turning to a radical treatment that sounds like science fiction. Here's Teresa Woodard.
There's such a high demand for this kind of new treatment.
Wow, so let's take a look.
Yeah, this is a clean room facility.
It was the first and likely only time for outsiders.
In a few weeks, we couldn't walk in here in street clothes.
Oh, absolutely not.
Got it.
Absolutely not.
To be allowed in.
Going to undergo a very deep cleaning of this whole facility, and then it'll be locked and sealed off.
Stephen Gray is aware of the fact
This is a bioreactor.
that his life's work sounds like science fiction.
We're manufacturing the virus here.
He will manufacture a virus in this lab.
Children are born, they have a defect where they're missing a gene, and then we put it back.
It is gene therapy.
500 liters.
Once injected into a human body, the virus he manufactures will deliver missing DNA
The air is going to get progressively cleaner.
in hopes of treating terrible diseases that missing DNA can cause. One disease is a lot like Lou Gehrig's, but in kids.
It's ultimately going to be, fatal probably by the second or third decade of life. I mean, what would you do if it's your kid? I have 3 kids. I mean, I would go to the end of the earth for them.
Come take a step. Come take another.
Talia Duff learned to walk. She learned to dance. This little girl with Down syndrome could stand and bike and hike. But instead of gaining strength with age, she lost it.
I'm tired. I know you are.
The name of this disease is CMT4J. Talia is now 13 and lives near Boston.
Well, it's like a neuromuscular disease.
Kinley Kitchens is 14 and lives in Texas.
You have like very bad balance and very lack of coordination. I fall a lot, so I have like a high tendency to like fall or trip.
Kinley's CMT4J is not as far along,
Hi.
but her family worries about the future.
The progression is what is just the unknown. You just don't know when it's going to progress.
Only 30 kids in the world
I'm hyperventilating.
have CMT4J.
Hi.
And in this room in Dallas,
How are you? Can I give you a hug?
2 of them came face to face.
How are you?
I didn't get it.
good. Dallas is home to UT Southwestern Medical Center, where clinical trials of Stephen Gray's gene therapy treatment are about to begin.
Does it feel kind of good to have someone else that needs a muscle medicine just like you do?
Oh, yes.
Muscle medicine is what Talia,
I like camera.
who clearly loves the camera, calls it. With Talia and Kenley and only 28 other CMT4J patients in the world, some question spending time and money on such rare diseases.
What's your favorite TV show?
But because there is no treatment,
Aladdin.
these families are willing to take incredible risks with potentially remarkable results.
And if we can do this for these rare diseases, then we can move on and we can expand the technology to things like Alzheimer's, autism, schizophrenia that are very common.
You like the cameras, don't you?
Reversing the damage already done isn't necessarily the goal.
It sounds science fiction, but it's happening.
If they keep the damage from getting worse, that's success.
It's so much hope.
It will mean their muscle medicine works.
This is going to be amazing.
In Dallas,
We've been looking forward to this for a while, haven't we?
Yes.
Yeah.
I'm Teresa Woodard.
Hope.
Conclusion. This study illustrates the ability to rationally design, test, and deploy a novel therapeutic agent for a patient with rare disease on the basis of an understanding of her specific pathogenic mutation. It is an example of individualized genomic medicine. It offers a possible template for the rapid development of patient-customized treatment.
ASOs are DNA or RNA-like substances, synthetic versions of those, that are able to potentially have or have demonstrated impact in a range of disease areas.
Antisense oligonucleotides are one of the 4 therapeutic modalities we use at Biogen. The most common one that people think of are small molecules. You know, like you might get an aspirin or something like that. That's a small molecule. Biologics are sort of the next wave of therapies that came along, and these are antibody therapies most frequently. Antisense oligonucleotides are almost a blend of a biologic and a small molecule. They're short synthetic structures that are very much like nucleic acids, but they're a sort of a gene-based modality. So now we can target very specific genes within the cell to treat a disease. Most of the remaining questions are, can this be translated into a drug? And I think that's what we're showing, is that you can successfully translate this modality that works so beautifully preclinically into something that actually has benefit to patients.
The impact of ASOs in the medical community has been significant. It's now offering a new and important treatment modality and treatment option that never previously existed. The introduction of therapeutic and approved ASOs in a number of diseases means that physicians now have a new option to consider for treating patients with a range of diseases.
This is one of the last great barriers in medicine. You know, how do you actually treat late onset neurodegenerative diseases? And this might be one of the key tools to doing that.